Now let's move on to look at type II hypersensitivities. This is a complex group of syndromes that involve complement-assisted lysis of cells by IgG and IgM. These antibodies are directed against those cell surface antigens. As you'll recall, cells have some unique surface markers that play roles in their transport, recognition, and development. These cell markers often have some significant differences between individuals, even though overall these cells are pretty similar between individuals. These molecules that differ from one cell to the other within the same species are called alloantigens, and they're often the target of this type II system. The reactions included in these type II hypersensitivities are things like transfusion reactions and some types of autoimmunities, as well as rejection of transplanted organs. Targeted cells are often the red blood cells, so much of our discussion in this section will concern red blood cells. These situations introduce alloantigens on the donor cells, and those are recognized by the recipient's leukocytes, so their immune system launches an attack against the donated cells. And this happens because the immune system's not really equipped to differentiate between desirable foreign cells and undesirable foreign cells that we see when microbes are invading. So the immune system is actually functioning perfectly well. It just cannot detect the difference between what we'd like it to accept and what it knows how to reject. Prior to 1904, blood transfusions were a really risky concept. Maybe it would work, but maybe not, and then you'd be dead. This is because sometimes the serum of one person causes the red blood cells of another to clump, because that serum contains antibodies to the foreign cell surface markers of the first person's blood. Study of this red blood cell clumping allowed us to come up with the ABO blood grouping system. Now, the ABO blood grouping is the most familiar of our blood types. However, there are about 20 other different cell surface antigens or cell surface markers on red blood cells, including the MN or the CAL and RH groups. But for the purposes of this discussion, we'll really be looking at the ABO and RH blood grouping systems. So in the ABO system, we have antigen markers on the red blood cells. These are genetically determined, and they're composed of glycoproteins. You'll probably recall much of this information from general biology, or at least from anatomy and physiology, but we'll go into the details again here so that you can refresh your understanding. So as you're probably familiar, within blood typing, we have a choice of A, B, AB, or O-type blood. These are the genotypes, or the allele combinations that could give this. So a type A individual could either have both type A alleles, or one type A and one type O allele, because as you'll recall, we get one chromosome from our mother and one chromosome from our father. So that gives us two chromosomes that will determine this red blood cell antigen label. So looking back at this figure, we can see that each blood cell type has this base on its antigen. Type A blood has an additional carbohydrate region, and type B has a different one. When we look at a type AB blood cell, it carries both of these antigen types in addition to the regular base subunit. However, type O blood only has the base portion of the antigen, or the common portion of the antigen. So the blood types, as you can see, are named for the dominant antigen. So A and B are both co-dominant. And so if A and B markers are on the cell, then it's type AB blood. If you have type A, you could be AA, which both are dominant, so A is displayed, or you could be AO. But because A is displayed on the cell's surface, then it has to be type A blood. Also keep in mind that type O red blood cells, although they don't have the A or B marker, they do have other antigens on their surface, because as you recall, there are many different blood typing systems. Also consider that tissues other than red blood cells carry these A and B antigens. So this is an important thing to consider. When blood cells are donated, it's the blood cell that's donated. The serum is kept separate, so we call them packed cells. The serum contains all kinds of other proteins, including antibodies. So a person with type A blood has serum that contains antibodies to type B blood, or anti-B. These antibodies will react with blood that's of another antigenic type. So the interesting thing is these antibodies are in the serum without any prior contact to the other blood type. Thus we call them preformed antibodies, because they're formed early in infancy, and it's thought that they're formed due to exposure to similar antigens that are widely distributed in nature. These antigens are things like surface molecules on bacteria and plant cells that mimic the AB antigens. So after exposure to these AB antigen mimics, the infant produces antibodies to those non-self foreign particles. The fact that there are preformed antibodies is what explains why there's such an immediate and intense transfusion reaction. These recipient antibodies attack the donor cells right away, and agglutination occurs. So here we have a type A donor, and these red blood cells, just the cells, have been donated to a type B recipient who has antibodies to type A blood. This is a situation that's going to turn out badly, because these antibodies are going to bind to the antigens on the cell surface of the red blood cells, and they're going to cause the red blood cells to clump or agglutinate. Now, along comes the complement system proteins. These complement system proteins, as you'll recall, literally act to punch holes in the cells. And so we'll see these red blood cells not only clump together, but lyse and release their contents. So let's take a quick look at this image of blood typing and consider the antigen-antibody interaction. This will help us determine whether we really understand what's going on here or not, and is the basis for blood typing tests. Now, if blood is not clumping, as in it has not been attacked by antibodies, it's going to look more like this, with all the cells evenly dispersed. However, if it agglutinates, as in the antibody is attacking the blood cell and causing them to clump, then we'll see more like this in a pattern. So here's some results from some blood typing cards. In this patch, we have antibodies to A blood cells. In this patch, antibodies to B. And in this patch, antibodies to RH. This is the one that gives us the plus or minus. So in this one, we have no agglutination, which means that there are no A-type antigens in here. Again, no clumping under B, so no type B antigens in here. However, we do see clumping under the RH factor, which means that there is RH, and so it's positive. So no A, no B, that gives us blood type O, but there is RH, so it's O positive. Here we can see that there's clumping under type A. This patch contains A antibodies, so if it clumps, that must mean that the A antigen was in there. There's no clumping here, so there's no B antigens, and there's no clumping here, so there's no RH factor. So that means we have A negative as the blood type. Check out these other two and see if you can understand how this is B positive or AB negative. So when transfusions are performed, prior to any actual transfusion, we'll take the donor's blood and mix it with the serum of the recipient to see if there's any sort of clumping reaction or agglutination. If there is, it's going to demonstrate some kind of incompatibility. Because remember, we're not just dealing with ABO blood typing. There are many other different types of cell surface antigens to consider when giving transfusions. So transfusions of the wrong blood type cause a differing degree of adverse reactions, the most severe of which results when the donated blood cells react with the recipient antibody and trigger the complement cascade, because you'll recall complement proteins assemble to make holes in cells, so they're going to lyse. This results in destruction of the red blood cells. So we have massive hemolysis, or breaking of red blood cells, that leads to systemic shock and kidney failure. We see the kidney failure because these red blood cell fragments will block the glomeruli in the kidney, and this is the whole filtering system. So often it can result in death. Less severe, we'll see fever, anemia, and jaundice. Anytime that there's an error in transfusion, we manage it immediately by halting the transfusion, administering drugs to remove hemoglobin from the blood because it's been lysed and released from cells, and then begin another transfusion with red blood cells of the correct cell type. Before we move on to look at Rh factors, let's take a quick look at a video on the type II hypersensitivity.
Type II hypersensitivities involve interactions of antibodies and surface antigens of cells, followed by complement-assisted lysis of these cells. A typical example is mismatched blood transfusions. There are 4 different blood groups based on types of antigens on the surfaces of the red blood cells. People who are type A have A antigens, and those who are type B have B antigens on the surfaces of their red blood cells. Persons who are type AB have both A and B antigens, and those who are type O have neither A nor B antigens. The serum of people with type A blood contains antibodies against type B antigens, and the serum of people with type B blood contains antibodies against type A antigens. Type AB serum contains neither antibody, and O serum contains antibodies against both A and B antigens. If blood from a person who is type B is transfused into a person who is type A, antibodies present in the type A blood react with the surface antigens on the incoming red blood cells. This leads to complement fixation and lysis of these cells. If blood from a person who is type A is transfused into a person who is type B, antibodies present in the type B blood react with the surface antigens on the incoming red blood cells. This also leads to complement fixation and cell lysis. Persons who are type O lack A and B antigens on the surfaces of their red blood cells and are therefore universal donors. Persons with type AB blood lack antibodies against A or B antigens and are therefore universal recipients.
Let's take a little closer look at this Rh factor and its clinical importance. The Rh factor is also known as the D antigen. So you can think of it as big D and little d if you like. The Rh type results from the combination of 2 possible alleles, just as the AB blood typing does. You can either inherit 1 Rh, 2 Rhs, or no Rhs. If you inherit 1, then we have to be Rh positive. If you inherit 2 copies of Rh, then we still are Rh. However, you could get 2 forms of the recessive gene, as in small d's, and they would be Rh negative. And the Rh blood type is unlike the ABO blood typing in that in order to develop antibodies against this antigen, you have to have sensitization to it, which occurs either through the placenta or through blood transfusions. The first exposure isn't such a big deal. This is a really important factor to consider in multiparous women, that is, women who have had more than 1 pregnancy. If a mother is Rh negative and her fetus is Rh negative, then there's no problem at all. If the mother is Rh positive and the fetus is Rh positive, then there's no problem either because both mother and fetus have the same blood type. However, if the mother is Rh negative, that means she does not have the antigen for Rh on her red blood cells. And if the fetus is Rh positive, then it does have the antigen on its red blood cells, which obviously he would have obtained from the father. And this difference in red blood cell type is no problem throughout pregnancy unless there are some serious complications. However, at the time of birth, we then have the mixing of the maternal and fetal blood. This also can occur during miscarriage. Now the mother has been exposed to the Rh positive antigen, and she will make antibodies to the Rh factor, which this time around is no big deal. But during the second pregnancy, she now has antibodies to the Rh factor. If the baby once again has Rh positive, then she will produce antibodies that attack the fetus. And this could be a serious problem. This is why we have RhoGAM shots. RhoGAM is an anti-Rh antibody. So for any mother that's had a previous delivery of an Rh positive or an unknown miscarriage, then she'll be given RhoGAM shots prior to birth so that she can receive this anti-Rh antibody that picks up all the Rh antibodies so that they no longer are capable of attacking the fetus and the pregnancy can go on without incident. Before we close this section, let's have a quick concept check. This one was from the animation. Which of these blood types is the universal donor? That means they can give packed blood cells to any other blood type individual. So a universal donor means that the red blood cells themselves have no labels on them, no antigens for which there are preformed antibodies in the recipient. Type A has an antigen on it, and so if it was injected into type B or type O individuals, then it would be agglutinated. Type B, the same story, and the same for AB. So type O in this situation is the universal donor. The universal recipient would be type AB because AB has neither A antibodies or B antibodies in their blood.